Hi there. Um, in this lecture, I'll be talking about some patterns of evolution and the evidence of evolutionary change. I already covered a little bit of this before, but this will be a little more comprehensive view of some of the evidence for evolution. Just a few definitions, and there's some distinction between the two of these, but they meld together eventually. But microevolution indicates changes in individuals within a population or species. So these are heritable traits that lead to new variations within a species or a population, etc. Macroevolution are these changes in individuals at or above the species level. So this is when you get formation of new species and you see new forms of the organism appearing. You can think of these conceptually as being on two different time scales. Microevolution essentially is happening all the time. You're having mutations within individuals. Those are leading to their progeny inheriting those mutations, and then those go throughout the population or the generations. Macroevolution happens more slowly on a longer time scale, so you can have speciation, which takes a number of generations to separate two different populations to become two separate species. For example, in Darwin's diagrams that were in the Origin of Species, you can see that for each generation there are microevolutionary events happening where you get a few of the individuals having a slightly different genotype. But then over longer time scales, these different individuals form new groups, new descendants, and those descendants form more descendants that have more variation. There's some separation, geographic or otherwise, and then you get speciation, which is the macroevolutionary aspect. So from Darwin's diagram of divergence, we can infer a few things. One, that there should be evidence of microevolutionary change, and we have demonstrated that over and over. And then evidence of speciation and macroevolution, that can be clearly demonstrated. And then evidence of common ancestry, and that can be demonstrated. And I'll show you a few pieces of evidence for each one of those. In his book, Origin of Species, he did discuss in Chapter 1 a lot about selective breeding and artificial selection, and that's direct evidence for microevolution. That is, you can get these changes within a species, within Canis, the dog, domestic dogs, and create new varieties from those, although they can all still breed, so it is a single species. You can also have direct observation of natural populations, this microevolution, so you can see variants within a population of plants, animals, or other organisms. And by comparative anatomy, that is, you can find vestigial organs that are still there in organisms that no longer need those organs, but their predecessors, their ancestors, did use those organs. So here's one bit of evidence for microevolution, that is selective breeding or artificial selection. So researchers chose two different female lines of mice. One group ran very little in the wheel during the day, and the others ran a lot more frequently per day. So then they allowed 24 generations of selective breeding. That is, they allowed the mice that didn't run as often to breed together, and the ones who ran faster to breed together. And then they measured how much the mice ran after those 24 generations. And the result was females from high runner lines ran almost 3 times as far as the control lines that usually ran slower. So it was a heritable trait. So they also asked the question, were they running more often, or were they just running faster? And so they looked at the number of revolutions and the time spent in the wheel for the two different groups. What they found was the high runner lines had neurological differences in which allowed them to run faster. So they weren't spending necessarily more time in the wheel, they were just running faster. And they looked at these genetic lines, and it actually mimicked ADHD, attention deficit hyperactivity disorder, in humans. So it did relate to humans as well. Another example is selective breeding that Darwin actually mentions in his book, and that's dog breeds. So dogs were selected from among wolves about 10 to 15,000 years ago, and it probably predated what we know as civilization, so it was probably more than 11,000 years ago. And the family tree comes from certain gray wolf lines, and then they were bred for different characters, and eventually we have the domestic dogs today. One of the initial and lasting characters, of course, was that they had to display some sort of domestic attributes. So one of the things was that we still see in dogs today is dogs usually want to please their owners. And wolves don't have this same characteristic, even if they're brought up in a human family, that they're much less to have that characteristic of wanting to please their owners. And this became an important attribute to modern day dogs in that they could be trained for different tasks and also to protect the family, etc., which was an attribute in many ways. It affected many types of behaviors in dogs. And today there are officially, hundreds of dog breeds in the world, recognized dog breeds, as well as anytime there's a litter of pups, you notice that there are differences even within that litter of pups. So that's an example of microevolution. Plants have also been bred for mainly for food, for fiber, for building materials, etc. One of the ones here is Brassica oleracea, and the wild plant looks quite different from the cultivated varieties. So all of these are the same species, just different varieties, including cabbage, where the vegetative parts are chosen for, Brussels sprouts, where the buds are chosen for, kohlrabi, the stem, kale, different types of leaves, broccoli, the stems and florets, and finally cauliflower, also florets. So these have all been bred for those specific characters. This creates some problems for the plant. For instance, cabbage forms such a tight head that it cannot flower without human help. So in order to get new seeds from the cabbage plants, they have to be split open so that the flower stalk can come up out of the bulk of the leaves. So the second part of this is direct observation of microevolution from natural populations. So the first example, which is in Darwin's chapter 2, talks about the inherited changes in field mustard, Brassica rapa, in response to changes in the environment. So whether it lives in a rainy area or a drier area, the days to first flowering, is shortened in the dry regions versus the rainy regions. So there's selection for that. And then when you produce hybrids of those two, the progeny are intermediate between the two types, the two ecotypes. This has been found in another plant, Arabidopsis. There are different ecotypes of Arabidopsis thaliana, and as you go up a mountain, what you find is that the ones that are lower on the mountain tend to be taller and they take longer to flower, whereas the ones further up on the mountain become shorter and shorter and have a shorter time to flowering, probably because it gets colder up there, so they have a shorter season to grow. Here's an example of microevolution that occurs in the soapberry bugs in Florida. The bugs use their beaks or proboscis to probe into the fruit to reach the seeds and feed on the seeds. But there's a flat-podded golden rain tree fruit that was imported, and the soapberry bugs that feed on that have shorter beaks. This was caused by the importation of this flat-podded golden rain tree fruit that was imported into Florida about 1926. So the soapberry bugs that migrated up into Florida from Key Largo, where they were feeding on the balloon vine fruit, initially had long beaks, but as time went on, there was no disadvantage to have a shorter beak, and so by the time 70, 80 years later, most of the soapberry bugs had short beaks for reaching the seeds of the flat-podded golden rain tree fruit. Both microevolution and macroevolution have been observed in the Galapagos finches. This one shows the microevolutionary events, which can be seen in the beak size of the birds, depending on whether there's a dry year when the seeds are then harder and a wet year when the seeds are softer. So the beak size actually does change from very robust, almost like a grosbeak would have, into more of a small, sharp beak when there are wetter years. Another example of microevolution is comparative anatomy, that is looking at vestigial structures in the organisms as well as their descendants. Here's one example in snakes. You can see vestigial structures in pythons and some other snakes, where they have tiny little leftovers of what once were legs in their ancestors. And if you do an anatomy of these, looking at the bone structure, you'll find that they do have the bones for the legs, but they're much reduced, and some are missing now. Here's an example in birds. This is the brown kiwi. It's a flightless bird from New Zealand. It does have tiny little wings, but they're completely useless. But this shows that their ancestors had wings that did work. Now, you also have other flightless birds such as emus and ostriches. Their wings are slightly larger, but completely useless for the weight of those birds. In apes, including chimpanzees and actually humans, they have a muscle that is an erector muscle that makes the hair stand up, and people still have this when they get goosebumps, right? These were made for displays when they were fighting one another, so we still have those as goosebumps. We don't use them anymore for aggression displays. And here's another one. Humans have a rudimentary tail, and some actually have longer tails as an abnormality, a mutation, which is common. But they no longer need a tail, but the genes actually are still there for making the tail from the ancestors that actually produced a tail. So moving on to macroevolution, the first thing I want to talk about are laboratory experiments, but I'll also talk about natural populations, the fossil record, and then evidence of common ancestry. So in Drosophila, which is a common laboratory insect or animal that's used, mating preference can be altered by feeding the organisms different food during their development. And so females that were grown on starch prefer males that also were grown on starch, whereas females that were fed maltose prefer males that were developed and grown on maltose. So this is starting to be a speciation event where these organisms can eventually become separate species that no longer are capable of mating with one another. And there are ample examples of looking at macroevolution in natural populations. One of the ones that's been recently demonstrated was over a period of less than 100 years, speciation was demonstrated in the finches on the Galapagos Islands. So speciation events don't necessarily have to take, thousands or millions of years. It can occur in less than 100 years. So here's one example in fish. The hypothesis with evolution is that you have a single variable population, eventually have distinguishable but interbreeding subpopulations, and then you get these subpopulations further separating with less and less interbreeding, and finally reproductively isolated populations, which equal species. And here's one example of stickleback, where one has longer gill rakers because it eats more plankton, and a second subpopulation in Canada that eats more bottom invertebrates, so it has the shorter gill rakers. When you make hybrids from these, which you can, the hybrids are sterile, so they are now two different species. Here's one example in birds of the greenish warbler. So the greenish warbler, the founding population, used to be in the Tibetan plateau, but then they migrated northwest and northeast to become two separate populations. And by the time they got back together, they could not interbreed, so they behaved as separate species. So this is one of the major ways that you get speciation, is you have geographic isolation, and then once they get back together, they cannot interbreed. So speciation, of course, happens in plants as well. They can do this by just slow variation followed by separation and then coming back together. Then they cannot interbreed. But one of the common ways that plants have evolved is to form hybrids and become polyploid. So in this case, there are two diploids that cross. So you have then a tetraploid is formed, and that tetraploid becomes fertile within itself, within that species. So it can happen quite rapidly, and about 50 to 75% of the flowering plants evolved by polyploidy. Other organisms did too, as well, including animals, but it's more common in plants. So the fossil record is full of evidence for macroevolution, including reptiles, dinosaurs, birds, humans, etc. And here's one I discussed in a previous lecture, where you have in South America fossils of glyptodonts that are the ancestors of the modern day armadillos. And there are several stages of these, so that you can find many examples of the same bones, same morphology, then evolving into what has become the armadillo. And in Australia, you also have ancestral wombats, some that are larger, and then becoming eventually the wombats that we see populating Australia today. Another one that I mentioned in a previous lecture was fossils of whale ancestors. So 50 million years ago, the ancestors of whales were walking on land, and so they needed front legs and back legs, as well as a tail for balance and for other purposes. By 38 million years ago, the descendants of the ancestors had moved into the water. So they had lost most of the appendages, the front legs and back legs, some becoming fins in the front and then the back legs becoming more or less useless in the back. And then finally you get to modern-day whales, where the front fins have elaborated more, and the back legs, what were the back legs, have completely disappeared. They're within the animal, although the vestigial bones, the pelvis, femur, etc., are still there, but completely non-functional. And here's a more detailed view presenting a phylogram with the various stages of evolution of those structures, starting with the ancestor that one line became the hippopotamus and the other line became the whales. First ancestors that walked on land, then those that were primarily aquatic or marine, and finally the ones that were completely aquatic or marine mammals. So through time you can see those front and back appendages changing, both the front legs, back legs, and tail, and the bone structures in those changing as well to reflect those changes from a terrestrial animal into a semi-aquatic animal, and then into a completely aquatic and marine animal. And then there's the evolution of the birds from dinosaur relatives. Archaeopteryx lived about 150 million years ago. That's sort of the middle part of when dinosaurs were on Earth. Dinosaurs lasted from about 200 million to 66 million years ago. And it had feathers. So the feathers in this case were not necessarily for flying, but mainly for gliding. And as I indicated before, they're about the size of a crow. Here is an artist's rendition of those. You can see that they have feathers, but the feathers are not made for flying, but more for gliding. You can also see it still has fingers on the ends of the wings and teeth in its mouth. And I think I mentioned last time, or in one of the previous videos, that modern-day birds do still have the genes to produce teeth, but they're turned off in modern birds, and they build a beak instead. The scales and feathers, all coming from the same types of skin tissue, can be traced through the different lineages by looking at fossils, parts of fossils. So you have the common ancestor of this dinosaur has primarily scales, and then you have rudimentary feathers, primarily to retain heat in colder climates. And then other feathers that became good for gliding. And then finally the modern powered flight feathers in modern birds. On a finer scale, these have been mapped out for different dinosaurs of the past. And you can see up at the top you have filaments and scales primarily in those dinosaurs. And then a later elaboration of plumule-like feathers that were primarily to keep the animal warm, pretty much like down feathers in modern-day birds. And then these pennaceous feathers eventually developed in the gliding and the flying organisms, including dinosaurs and birds. Viewed in a little more detail, these evolved from simple filaments to tuft-like feathers, and then tufts with barbs and secondary feathers. And then some of these barbs became intertwined, and those are the flight feathers that even in the powered stroke of the bird, the individual filaments don't come apart because they're essentially knitted together by these fine filaments and side filaments. So looking at the evolution of these different structures, starting from skin, which is the simplest form of these epidermal cells, they can be folded over to become a nail or a hook, or a claw. They can be folded inward to become glands, and then they can start forming things like feathers, tufts, more elaborate feathers, including feathers primarily for heat retention, those for gliding, and those for flight, powered flight. And so these all come from the same types of tissues, but have been elaborated during evolution. So this diagram is a phylogram of those different organisms and showing a few different types of genes, including keratin genes, patterning genes for where those genes are turned on, and then some other genes relating to specifically to feather formation. So you can see that the genes for patterning of where to put different structures on the skin were there and in multiple copies very early, but the keratin genes, which form hairs and feathers, didn't appear until later, and so you finally find those appearing in in many more copies and being expressed at higher levels in dinosaurs and birds, and to a lesser extent in mammals. So this was actually an interesting study because it showed that all of the genes necessary for making feathers and for placing feathers on the body were there long before there were feathers. But then through evolution, the formation of feathers was more and more selected for, and so all of those genes that were already there were just made into more and more copies and were expressed at higher levels to become those hairs and feathers. So essentially, this is one of the things that you often find in evolution, is that in this case, before there were feathers, the genes to make feathers, to finally make feathers, were already there long before they were necessary in those organisms. The final piece of evidence for macroevolution is evidence of common ancestry, homologies of structure, etc., and evolutionary and phylogenetic trees, tying things together, using gene sequences usually. So one example is looking at comparative anatomy. So if you label each of the bones in the human arm and hand, you can find those same bones in the mole, although arranged differently in different sizes, in the horse, in a dolphin, and in a bat. So this indicates that these structures have been in these organisms for millions of years, but they've elaborated in different ways in those different organisms. Reproductive structures are often fairly well conserved during evolution because you have to have these reproductive structures to carry those genes forward to the next generation. So if you look at the structural homologies within these plants, you have petals, anthers, stigmas, and although they can be quite different morphologically, they serve exactly the same purpose in all of those flowers. If you look at development, there are many homologies during development, and this was first elaborated by Ernst Haeckel in the late 19th century. So if you look at snake, chicken, possum, cat, bat, humans, all vertebrates, right? You can see some, in early development, some homologous structures, and in some cases, if I gave you one or the other, you would be not able to tell which one is which. And then in the middle of development, there are a few more differences that you can see and that develop. And then finally, a late form during development, you can finally see some of the structures that distinguish one type of organism from another. There are sets of genes in all vertebrates that are known to be common to all vertebrates that occur in early to mid embryogenesis that determine that organism will be a vertebrate organism Later in development, they actually have a wider variety of genes, and that's what makes each one of those organisms, those species, develop the unique traits, the morphological traits that they have. So in Darwin's tree, then he indicated that there was evidence of common ancestry, and this was primarily based on morphology. So you would look at one organism and determine that a fossil organism was related to that one by morphological characteristics, and so that there was descent with some modification. That is, there was variability or diversity within each generation. So here's a little hypothetical example of that, where you have snails starting with an ancestor, and then at some point in time there's a variant with elongated shells, and then a variant with bands. Another line becomes pink, so everything beyond that is pink. One of those branches attains high spires, and so you get these ones over here with high spires, and then one group has a variant that develops spikes, and so you can line these up in a logical fashion to show how evolution has proceeded in those organisms. You can see this in cats also. 8 species of cats running from Canada lynx to leopard over here. And you can go back to the most recent common ancestors had spots. Some of those retained their spots, although they had a bob tail then. You get the bobcat and the lynx on one side. Further up, some of the descendants lost the coloration, so they had not spots anymore, but a uniform coat. And then you get rosettes to form leopards and snow leopards and other types of leopards. And then along one lineage, those spots have sort of joined together in stripes to form the stripes on a tiger, and some cats as well, some domestic cats. So you can link these together in a tree to form, to show that those forms, those spots, have changed through different lineages along that phylogenetic tree. If you look at other genes also, it's consistent with this branching pattern, so that then you can determine, what sort of patterns have occurred in the past and how they've evolved. These patterns are sort of interesting, by the way, in that not only one layer of skin cells forms the pigment, but they're turned on in different ways in different layers, so that you can get these different types of patterns. So it's much more complex than you can even show here. So here's just a summary for the evidence for evolution. On a small scale, evolution can be observed in nature. So this is microevolution. Evolution can be produced experimentally. Newly reproductively distinct species can be produced experimentally. They can be observed in nature as well. Fossil evidence shows the transformation of species and speciation events. The order of the main groups can be determined from the fossil record. And you can see a hierarchical organization of life and what is what we now call species. And finally, homologous similarities can be found among groups of living organisms as well as those from extinct organisms. Okay, so that's the end of this part, and talk to you later.